Hello bachcho, let us look into the question. The steps in DNA fingerprinting are these follows, and we have to tell whether what are these variables A, B, C, D, E. Let us look into it. The key concept is fingerprinting, that is DNA fingerprinting. This technique was developed by Alex Jeffrey in 1984, and fingerprinting is based upon the principle of polymorphism of satellite DNA. Under satellite DNA, in DNA fingerprinting, we study the trends of VNTR, which is variable number tandem repeat. Apart from that, when we talk about the process of DNA fingerprinting, it involves isolation of DNA, digestion of DNA, isolation of DNA from the genome, then digestion of DNA is done by restriction endonuclease. So A will be restriction endonuclease. It worked over the palindromic sequences and it is used to incise the DNA, cut DNA into fragments. Separation of DNA by electrophoresis, that means by gel electrophoresis on agarose gel. Agarose gel पे इलेक्ट्रोफोरेसिस कैरी आउट किया जाता है जिसमें कि डीएनए फ्रेगमेंट्स को अलग अलग सेपरेट किया जाता है on the basis of their size. Apart from that, when we will be talking about the next statement, it talks about transferring of DNA to. Now C is transferring of DNA to the membrane, which is nitrocellulose or nylon, that is a membrane. So it will be nitrocellulose or nylon membrane. Since it is a process of sudden blotting, that means isolation of DNA, so it will be transferred from gel to membrane. Apart from that, D की बात करें, it is DNA hybridization using labeled VNTR probe. Labeled VNTR probe. They are short DNA sequences that are rich in GC repeats. They comes under mini satellite that possesses more than 5 base pair. And when we talk about VNTR, it consists of 16 to 64 base pairs. Now we will be talking about E-Autoradiography or X-ray sheet. Through this audio radiography, what we see that detection of hybridized DNA is carried out. So these are the answers, and we can say the correct option is A. Option A is the final answer. Hope you all understood. All the best.